Good morning, everyone. It is a pleasure to share the update from the DERMAL working group. I will share about our mission, our progress, and the different steps. So you will see that this presentation summarizes where we are today and where we're going. The DERMAL working group is the most recent working group. So you will also see that we are at an earlier stage of the development, but progressing relatively rapidly thanks to the understanding and the development that was done by previous groups. You will also see that we have selected a different set of botanicals, and we have updated that list that includes different type of extract and essential oils.
Cecile, I still see you on the first slide.
Okay, sorry.
No worries. There we go, there's the mission, perfect.
There we go. So our mission is straightforward and ambitious at the same time. Our aim is to develop a screening strategy that can reliably identify potential dermal phototoxicity, irritation, and skin sensitization for botanicals as complex mixtures. And the key challenge, as you know, is that botanicals are complex mixtures. Here we go. So we are fortunate to have a truly interdisciplinary team representing government agencies, academia, and industry. As you can see, we have companies that both provide testing methods as well as companies using the different botanicals. Here is an overview of the workflow. Briefly, we start by identifying botanical cases. We are selecting plants that will provide different responses in the different tests. So here we are focusing on phototoxicity, skin irritation, and skin sensitization. We are then selecting OECD guidance tests that have been proven to work for single compounds. And now we are in the phase of the next steps, where we are testing the botanicals for their constituents and in the different tests. And next, our goal is to determine if the existing OECD guidance are performed according to what we expect, and then the results will be published. Here's the list of botanicals that we have selected. Our selection process was focused to identify botanicals that represented a range of complexity and usage profile and potential risk factors. So here in the list, you can see that the first three were selected for their negative effect or no effect. The second two were selected for their potential sensitization, then the next three for irritation, and then phototoxicity. We have also selected a botanical that was expected not to be phototoxic based on the absence of furocomerins, which is the bergamot-free furocomerin, the furocomerin-free bergamot essential oil. Here I would like to point out that as well that we have selected different types of extracts, CO2 extract, ethanol extract, as well as essential oils. Now onto the assays. So our botanical materials are selected and characterized. Then we move on to the evaluation phase. And these are the different tests that we have selected for phototoxicity, skin irritation, and skin sensitization. These assays are all OECD recognized and form the foundation for non-animal safety testing. For phototoxicity, we are using the OECD 432 in vitro assay, and as well the 498 reconstituted human epidermis phototoxicity test. These methods evaluate how a substance affects cell viability in the presence and absence of UV light. For irritation, we are using the OECD 439 reconstituted human epidermis test This model mimics the architecture of the human skin and allows to measure cell viability and the inflammatory response. Finally, for skin sensitization, we are aligning with the updated OECD guidance 497 that includes different methods. And here we are focusing on the three key events, 1, 2, and 3. Key event 1 focuses on protein binding, while key event 2 on keratinocyte activation, and key event 3 on dendritic cell signaling. So for each assay, if we start here with the phototoxicity assay, the test is done in mouse fibroblast cells, and the cells are exposed to UV lights, and then we are reading the concentration. Sorry. The cytotoxicity is dependent, is proportional of the uptake, or inversely proportional of the uptake of the neutral red, sorry. For skin irritation, we are using the OECD 439, and in that method, cell viability is measured by enzymatic concentration of the MTT that is converted in the blue formycin salt. Here we can quantify also the cell viability. Now for skin sensitization. This is the most complex endpoint. It refers to the process by which a substance triggers an allergic contact dermatitis response after repeated exposure. It is a type 4 hypersensitivity reaction. It is mediated by T cells, and it follows a well-known set of events. They are called adverse outcome pathways. And each test follows or focus on one of these key events. The DPRA is designed to detect skin chemical binding to skin proteins, while the keratinocyte activity is measured by the keratinosense, which captures inflammation responses. This is the key event 2. Finally, the dendritic cell activation, also referred to key event 3, is modeled in the human cell line activation test, or the HCLAT. So by combining these 3 different methods, we are able to evaluate how sensitization, how a compound may be sensitizing. Finally, it's important to look at our different highlights of this year. We have selected and sourced our first set of botanicals. We have launched our chemical characterization and the testing. We are drafting our safety paper, strategy paper, sorry. We are exploring in silico methods. And also it's worth to mention, we have been invited to a full submission of a grant. I don't have a slide to show our outreach initiatives, but we had a poster at ICSB, and we now have a draft abstract for SOT. I would like to thank you all, and I would like to take questions. Thank you.
There we go. My video wouldn't come back. Thank you so much, Cecile. That was a really excellent presentation. It's always a little bit trickier when we're planning, but we have so much that's going to be coming in the next year. So I think that's really exciting. Cecile, in your own work at doTERRA, you've done some work with skin sensitization in assays. Are there any watch outs you've seen for adapting the assays with essential oils, or so far have they worked sort of as is?
Um, that's a good question, and thank you for asking that. Um, with essential oils, there's a solubility that is an issue. We also have the volatility to manage.
Yeah.
So although we haven't at this point really know how to adapt this, we definitely is something that we're looking into.
Thanks for that, Cecile. There had been a hand that went up during your talk. I wasn't sure if someone accidentally clicked a button. If anyone does have another question or thoughts, they can let me know. Angela asked, have you selected only botanicals of topical use for dermal toxicity? Could you speak to that, Cecile?
We have selected botanicals that some that are used and some are not used, and I don't know where the question was it on whether they are used or not. They are not necessarily used in the industry. However, some do provide good controls as positive controls for the non-irritation effects or sensitization effect or even phototoxicity. But yes, some of these botanicals are used in the industry.
Yeah, thanks, Cecile. I think that's a good point to emphasize too. Hopefully no one's using poison ivy in a product.
Poison ivy is one of the examples where that is not used. Another one, the other one is cinnamon. It is used and is known to be sensitizing. So it's usually recommended to be diluted and so on. For the others, we have also negative controls.
Mm-hmm. I think what Angela was maybe asking is, were these selected primarily for their dermal, like their topical use versus like oral or another route?
Some of them might be used internally, but we are in this test evaluating the sensitization, irritation, phototoxicity for topical use.
Great. And then one other question, are you considering the use of any 3D skin layer test, either at this time or for future assessment?
That's a good question. We are in evaluating or looking into in silico methods at the moment. We haven't. I don't think we have discussed that yet, but that could potentially be a next step in our evaluation.
Yeah, I know we kind of looked at what tools people regularly use, but there are more like 3D ones coming on. I think that could be interesting to explore once we launch this future phase, like this initial phase of work for future. We heard from some of the other presentations, some of these 3D models might be a little more human relevant or have additional cell types, but I'm not as familiar with skin models available.
Yes, 3D for skin. I don't know either.
That's okay.
There's epidermis, reconstituted epidermis that we're already using. So it's, I think, already a good model in this way.
Great. Thanks, Cecile. And William, who asked the question, if you have any info on that, please feel free to send it our way. I think that would be interesting to explore. Okay, with that, Cecile, I really thank you for your presentation and your work on all the different groups. Julie, I'll turn back to you to present Amy.
Great. Thank you, Cecile and Connie. So our last presentation of the morning, but certainly not least, will be presented by Dr. Amy Rowe from Procter & Gamble, who will be presenting on behalf of the Hepatotoxicity Working Group.
Okay, great. Thanks, Julie. Can everyone see my screen? Did I share?
I do not see your screen, but I hear you.
Okay, great. Well, let me go back and try again. Okay. Share screen, entire screen.
Yeah.
Okay. Now I'm sharing, I think.
Yeah, we see the web app right now. Oh, there we go. Now we see slides. One step at a time. Yeah, perfect.
Got it? Okay, great. Thanks. Okay. Well, we're in the home stretch, so it is my pleasure to present the progress from the hepatotoxicity working group. I am presenting today in lieu of my co-chair, Dr. Steve. Sorry, I'm having a brain moment here. Steve Ferguson, due to the government shutdown, obviously he was unable to present. So really sorry for that. Our mission is the same as everyone else, so I'm not going to spend too much time on that. Here's a list of our current members. As I mentioned, Steve and I co-chair this working group. And like all of the other working groups, we have membership from industry, academia, and government. And I won't, go over the list in too much detail. We started with the original 13, and what you see highlighted in green here is, reports in the literature, either in vitro or in vivo, in animals or human case reports of liver liability, liver toxicity, or potential botanical drug interaction. And so, some of the data is, maybe more solid than others, but nonetheless, we wanted to explore what we saw in the screening tools that we chose for looking at hepatotoxicity. And so we are really fortunate, I think, in the hepatotoxicity working group that we have two models involving hepatocytes, 3D hepatocytes and 2D hepatocytes. We're focusing just on human hepatocytes, but of course, hepatocytes are available from multiple species. But these models have been used for, in some cases, with the 2D for decades. And so we have tremendous amount of historical knowledge, both in terms of, chemical data, chemical testing data. Some, probably more than maybe some of the other assays, complex mixtures, including botanicals. But also some of the tools, the screening tools that we're using, they've just been used forever. So we're fortunate for that. And we are looking at, on the right-hand side, we're looking at a number of assays. Some of them we're looking consistently between 3D and 2D. And everything from just your basic cytotoxicity markers, ATP depletion, LDH release, other markers of cytotoxicity, which you would perform in any anything you're testing. You always start with cytotoxicity in order to delineate the appropriate dose response curve that you want to use. But we're also including more modern techniques like the transcriptomics and cell painting to look at effects on cell structure. So those are, you've heard some of the other working groups are using transcriptomics, but we're using it as well. And so that's a newer kind of screening tool that we're anxious to see how it works. So I'm going to start just presenting some cell viability data for the 2D hepatocytes. And this work was done by Dr. Albert Lee. And of course, certainly an expert in 2D human hepatocytes. So in the top pane, you'll see LDH release data for both, which I'm just showing you milk thistle and kava here. Milk thistle as our negative control, kava as a botanical where there have been some reports of potential hepatotoxicity. And then in the lower pane, we're looking at an MT glow assay, which is a way of measuring cell viability. And what you see across this broad range, very broad range of concentrations, for milk thistle, a little less so for kava, is that, we do, although the data look a little little messy. You know, what we see is certainly a trend in increasing cytotoxicity with the you know, higher dose levels, and that drop off that you see at the end at the highest dose level is pretty typical when you get to very high dose levels and you start to just have dead cells. You kill cells, and so that's not too uncommon. You see this consistent between the LDH release in the top pane and the MT glow in the bottom pane. A little less toxicity, cytotoxicity for kava across the dose ranges that we tested. And, you know, just to kind of, you know, proactively address the question about how we chose concentrations, you know, similar to what others have suggested. You know, at the onset, we wanted to test the broadest concentration range that we could, and then, you know, have data available to then go in and look more specifically at how these levels correspond to what's used in dietary supplements. And so I would kind of refer back, if you can recall, to, I think, Catherine's response to that question from the DART working group. You know, it's a very similar approach that we will take. So next, we'll look at cell viability in 3D hepatocytes. And we see, you know, very similar profile. It's not really until you get at that higher dose level that you see the LDH release. You know, a similar profile. You know, milk thistle seems to be more sensitive at that. We're testing a much higher dose range than what we could test in kava. Oftentimes, we were just limited by solubility in terms of how high we tested. We tried to test to the limit of solubility. So next, we move on to looking at cytochrome P450 inhibition and induction. We'll start with the 2D hepatocytes. And we only looked at 3A4. And that's primarily because we really just, you know, from a resource limiting standpoint, you know, we chose to look at one P450 to begin with, 3A4 being the one that's probably most involved in, you know, of course, drug metabolism, and has the probably highest levels in liver. We chose to look at 3A4. And so with induction, we did not see any induction across the dose range that we tested with milk thistle. So that is not unexpected. That was we thought would be an appropriate negative control. But we saw a really strong induction response with kava, a stronger response, in fact, than what you see with St. John's wort, the far right-hand pane. And St. John's wort, of course, is known to be a positive control for P450 induction, 3A4 induction. And so this was kind of a surprise to us. There's not a lot of hepatocyte induction data for botanicals. Actually, you know, most people, you know, just don't go that far. So we hadn't seen this before, and we're quite interested in these results. And now looking at inhibition, we again are showing milk thistle and kava here, and looking at 3A4 inhibition, and we really see no results. No inhibition. You see a decrease in the viability for milk thistle, but that's really due to the cytotoxicity that I showed in that early slide. So no inhibition from either milk thistle or kava, but definitely induction from kava. Now, moving on to the 3D primary human hepatocytes and looking at inhibition, we see a similar profile for inhibition. No inhibition from either milk thistle or kava. Maybe a slight inhibition at the upper treatment level, but again, you're starting to see a little cytotoxicity. We used ritonavir as a drug positive control, a known potent inhibitor. And so, you can see what really potent inhibition looks like. So we try to use botanical positive controls when we can, but we also use, drug controls because of just that broad and historical knowledge that we have in hepatocyte models. Now, Steve also looks at induction using a transcriptomics approach. And so he's looking at various genes for things like PXR, CAR, and the regulatory mechanism of induction. And what you see here is an interesting way to just look at potency of induction and of based on gene expression. So both milk thistle and kava cause clear changes in some of those genes that he was looking at. Milk thistle in blue there on the far right hand side obviously was the least potent of the botanicals that we looked at. That is as expected. And then kava was the most potent, as you can see in red. And you'll see data here from Yohimbe. I'm not speaking in depth on that, but we also see some interesting induction with Yohimbe, and so we're following up on that as well as a case study. So this provides some potential mechanism of action information as well. So and makes it for really easy way to kind of get an overall picture of potency and maybe help one to prioritize what you might want to follow up on in more definitive assays. And then lastly, I'd like to present just briefly some work. We also are interested in looking at potential reactive metabolite formation. And this is just a brief slide on work conducted by Dr. Richard Van Breemen's laboratory at Oregon State University. And he, incubated kava with human liver microsomes, either with or without human liver microsomes, and showed the formation of glutathione conjugates with both milk thistle and kava. And so you see the conjugate with the phytochemical constituent taxofolin, or some of the metabolites of kava lactones. And so, again, this may help address some of the mechanism of action of some of the liver toxicity that one might see once glutathione is overwhelmed and there's potential for reactive metabolites. So in discussion, we think that the liver enzyme leakage, the LDH release, worked well in 2D cells for botanicals. And this is, probably not too surprising. I mean, this, I've certainly used LDH release in a number of studies that I've conducted personally with various botanicals. Steve felt that the 3D models, are less sensitive to cytotoxicity with these botanicals. Kava induced 3A4 at levels comparable or higher, actually, than St. John's wort. And so we really want to follow up with that to see if the results are clinically relevant. So, we have various ideas of some work that we, might like to do. And, if funding was available, we'd even like to do a clinical interaction study if we could, just because, this hasn't been shown. Omics approaches and reactive metabolite data have been generated, and we think that those approaches can aid in providing mechanism of action insights. And like everyone else mentioned, we're ready to now begin, really looking at how we can use ADME data to, better position the data, understand in vitro to in vivo extrapolation, clinical relevance of some of our findings. And we're also excited, that work has been launched with some advanced liver models with a few of our botanicals. This is being done at FDA by Dr. Kirsten Ekstrom, who's a member of our group. And so we're excited to see the results from her work. I think, as others have mentioned, we've started to look at some constituents. We'd love to follow up on further on some of the constituents of some of the botanicals, particularly where we're seeing some interesting results, and test those. And we have a number of manuscripts in preparation. This is a manuscript that we put out some time ago. And then we also had a session 2 years ago at the Society of Toxicology, really focused on botanical induced toxicity and liver injury and botanical drug interactions. And it was really well received. Had a really nice attendance and a lot of discussion from the audience. Obviously, a lot of interest in these endpoints, and so we published that as a workshop report. We presented some of these data at the recent SOT and the ICSB meeting, and we've also presented the data to the FDA in a presentation that Connie and I gave earlier this fall. And we are considering a number of next steps. Obviously, we like others, we're trying to determine what do we do with all of these data that we've generated. Too much, as Christine said, I think too much for a single publication. So we're looking at how we parse out the data into multiple publications. Do we, some of the data on just a single botanical are probably interesting enough for a publication. Like others, we want to collaborate with other groups, and also looking at future work. Understanding the clinical relevance, of course, is key, especially with botanical drug interactions. I think, we see a lot of reports of in vitro interaction potential, but it often doesn't translate to clinical relevance. And we want to look at potentially some additional cell models. We've talked about organ on a chip. And one of the things that's not listed here, but many of our members are interested in incorporating gut models into testing. And so we've actually, reached out to a few groups to understand, their gut models and how we could incorporate, the gut and the liver, because I think it's really key to understand, the two together. And then, as with others, lot-to-lot comparisons of botanicals to really understand assay responses. So with that, I will stop sharing. And I think we're ahead of the game. And so
Great. Thank you, Amy. We have one question in the Q&A already. So with regards to the transcriptomic data, have they been used to look at only at the single genes? Has any pathway level analysis been performed? Specifically considering there are AOPs for liver toxicity, has there been any attempt to look at whether the transcriptomic profile provide evidence for the presence of key events that may suggest downstream adverse effects?
Yeah, no, we really haven't gotten to that depth yet. Clearly, that's something that we want to do. And in fact, we've really only looked at just a very brief look at the three botanicals I think that I presented. That is one of the things that we were actually hoping to begin to deep dive on in our day two of what we had hoped to be day two of this annual meeting as a working group to really deep dive into the data there. But I think really dependent on Steve to help lead that, and obviously with the situation that's going on, we were unable to make that happen. But maybe whenever we get that day two rescheduled, we'll be able to focus in on that. But clearly, absolute great suggestion and where we need to go.
Great. Thank you, Amy. We have another question. Is it possible to determine which constituent of botanical plays the primary role in CYP inhibition or induction? Is it also valid to investigate whether there are interactions between different constituents?
Yeah, well, so, I mean, certainly I think it's possible, maybe quite labor intensive, but definitely I think, we will start to, in some cases where we're particularly, we have interesting results, say for the kava induction results, for example. If we wanted to really try to understand what is the phytochemical constituent or constituents that's primarily responsible for that induction, we could certainly start to look at that. But you can imagine it becomes quite complex, right? Because do you just choose the phytochemical constituents at the highest concentration in the extract to look at, or, who's to say that maybe it's a minor constituent that's the primarily involved in the finding that you're seeing. So that can be a little bit tricky, but that is something that we are interested in trying to do some work on. So any suggestions that anybody has, again, I mean, these are, I think, a great way for those of you, participating who are maybe not currently actively involved in the Botanical Safety Consortium. There's always ways to get involved with some of these ideas.
Great. Thank you. Are there any additional questions? Feel free to raise your hand or put them in the chat.
I guess I would, just to, kind of build on that as I'm thinking, I think the next step, right, is to really start to incorporate that ADME and understand the relevance of the concentration range of where we see those findings to determine, are we just, so much higher than what is really realistically used? Would we even bother to go down that path? So I think that's the next thing we need to think about is just, how do we determine whether or not to pursue some of these additional studies. So I think that's probably what needs to happen next is just the relevance piece.
Great.
May I
Thanks,
had
Amy.
a thought and want to get your opinion on, for hepatotoxicity, there's so many cell types, right? And there's so many cell setups.
Yeah.
We probably can't do everything, but could you maybe speak at a high level on like Hep G2 versus Hep RG versus the work we did in primary human hepatocyte?
Yeah. Well, I think we really like the human hepats, the primary cells, right? We like the primary cells because we have this historical body of knowledge. They're used. They're the gold standard, certainly in the pharma industry. And so we really like to use those cells. So I would kind of hate to utilize our time and resources towards other, cell models that aren't primary, unless it was to help maybe in work with other working groups who are using some of those cells, if there was that collaboration that could be made. But you reminded me of, something else I wanted to mention. And that's another thing that we're considering. And we've talked to folks who have, you're right. The liver is very heterogeneous. I mean, we're kind of biased towards hepatocytes, but there's many other cells that are there, particularly Kupffer cells, the resident macrophages, the endothelial cells. So that's another thing that we've really considered, is looking at co-cultures of primary hepatocytes with other cells, especially so much of liver toxicity may be immune mediated. So I think that becomes very important.
Great. Thank you. Alrighty. So I believe we're roughly on time to move on to open discussion and wrap up. So while you all think of some potential discussion points, we have a Menti poll that we can go through. And I think Connie might do a demo of how to log into the Menti poll. Just to, it'll probably be easier to do on your computers than phones.
Yeah, you can do either, but what you can do, and Julie will hopefully put the link and the code in the chat while I'm doing this. So you can copy and paste the link or just go to menti.com. So you go there, and this is a polling website. Some of you might have used it before. Hopefully you can see it on my screen. And then you're going to enter a code. So I'm going to copy and paste the code that Julie just put in, the 67691. So I'm going to put that there. And then once you're there, you'll basically be told to wait a second, that you're there already. But hopefully we can get folks to go there. You can do it on your phone. You can do it on the computer. It might be easier on the computer. So I can, hopefully I was sharing, so I can stop sharing, Julie, now.
You were sharing, and thank you, Connie, for the demo. Okay, so now you should be seeing the intro slide to the Menti on my screen. I see people are logging in currently. We're going to have a couple like warm-up poll questions while people are logging in, just to get familiar with the platform. And then we'll go into some more open answers on next steps for the consortium. I'm going to wait till the participant number maybe stabilizes a bit. I see we have 82 attendees. We're at 30 participants, but slowly climbing.
You can also do the little heart as you wait, or a thumbs up, or a devil, or a cat. I don't know what that one is.
I thought it was a cat. That's why I added it.
Oh, okay. I think you're right. Awesome.
I'm sure our working group members know I have cats, and their tails will come by every now and then, so it seemed appropriate. And if anyone has any questions or difficulties, feel free to reach out to Connie and I in the chat for the Zoom.
Yeah, you might have to do the Q&A, but we'll get it.
Yeah. Whichever way you can find.
That might be good to at least get
right.
rolling as people get it. Yeah, 45. That's pretty good.
Yep. Okay. So our first sort of warm-up poll question is, what best describes your perspective in the consortium? So industry, government, academia, nonprofit, or other?
We're also, I do want to say, and people have mentioned with the government shutdown, we've lost about a third of our participants today.
Yeah.
So it might not be shocking that we have a 50, 60% industry today. And we might have government from other jurisdictions, which is awesome that we still do have that participation. But I think it might be slightly skewed today just because of the shutdown.
Yeah, as I was putting it together, I was thinking of that. Great. So still pretty good representation given that the US government is currently shut down and many people weren't able to join. So we'll go to the next sort of warm-up question. So which of the following best describes your level of engagement with the Botanical Safety Consortium? So the options are active member of one or more working groups, a BSC member but not currently active in a specific working group, not a member but regularly follow BSC activities or updates. So I know some people come to our annual meetings almost every year, and then new to the BSC / first time attending a annual meeting. So it's nice to see lots of our working group members were able to join. I know we've been reminding you all about the meeting for probably 4 or 5 months now. And a good number of non-members, but who frequently follow the BSC activities, which is nice to see. It makes me feel like our outreach is validated. Great. All right, so now that we've done the warm-up questions, everyone should be a little bit more familiar with the Menti platform. We're going to go into some open answer questions. So the first one is, which areas should the BSC prioritize for further work? More botanicals?
Yeah, like Julie said, this can be open-ended, but if you're like, oh, we really need to test 50 botanicals, that's it. Or if we need a renal toxicity group, that we don't have one of those, or inhalation, this is where we really would like to hear from you all.
So botanical drug combinations, guidance on determining botanical constituents. I know one topic that's come up a lot in our working groups is, and I think was discussed in almost all of the presentations today, was like lot to lot comparisons and seeing how the different lots respond in the assays. So I'm going to hit space. And theoretically, AI will magically start grouping our answers.
This will show if we still need me and Julie or if we can just AI everything.
Yeah. Some more botanicals. We have about 8 responses. In vitro, which is a pretty broad category, Menti, but if we click on them, so it seems like sort of IVE, so the in vitro to in vivo extrapolation. Constituents. Oh, we have more responses that weren't categorized. So if I hit space again, nope, it doesn't want to redo it.
Maybe we are
It's too early. Yeah. Potential use of botanicals in cosmetics, mechanistic insights. Great.
I
And we'll
like the
be able to
combo botanical one. That was interesting. That's something we've talked about, like, multiple botanicals. That's like a whole nother level of complexity. That's cool.
Yeah. Yeah, and I know if you look at some of the botanical products available on the market, there'll be like a combination of 10 different botanicals to look at. And Connie and I will have all of these individual results, so they'll help guide next steps potentially for the consortium.
Yeah.
All right, so, oh, you can vote if you want. I don't know if we need that. So we'll just go to the next slide. So one question we had is, where could cross-working group efforts add the most value? So I know Shane mentioned potentially like cross-working group efforts between the neurotoxicity working group and the cardiotoxicity working groups due to some similarities in the signaling. So we were wondering what other cross-working group efforts could be beneficial. Yeah, add me with pretty much all other groups, especially with these in vitro methods. And that's related, I think, to the IVIVE as well. genotoxicity and hepatotoxicity, analytical with all others.
Yeah, one thing we didn't really talk about today, but it's come up in probably every work group over the years is, for some of the assays, could we almost keep the cells or the test system and then quantify key constituents that are actually in there to see what's getting in? It's not something we've done so far. I think it would be really cool, but a little bit tricky. I mean, but then if we already know what markers to look for, maybe we could do it in select cases.
Yeah.
Ooh, metabolomics. Folks want to know I also manage our omics work, so I get excited about that.
Let's see. So core battery, so comparison of neuro, cardiac, respiratory. We don't currently have a respiratory group, but that could be something to expand to. IVE.
Yeah, some of these are like gene tox start, if something is potentially causing genotoxicity, you think it might disrupt how the cell is developing and going. So that might be really cool. Probably gene tox
Yeah.
in a lot of these categories too.
Yeah. Much like ADME in a lot of the categories.
Very cool.
Yeah. See if we have any trends from the about 20 answers we've gotten so far.
ADME, gene
Yep.
tox,
Transcriptomics, IVIVE,
analytical.
metabolomics. Cool. This will be really helpful. I'm gonna skip through the voting again. So this one I believe should be a word cloud. So it is which technical challenge most limits the practical use of NAMs for botanical extracts? So it could be, yes, solubility. I know this is one that's come up in quite a few of our working groups. The word clouds are kind of fun. I know another concern was potential matrix effects or binding of active constituents to the actual test systems themselves. That's always a concern for some of the new approach methods that are done in the plastic well plates.
Yeah, I like the lot to lot comments as well, because you all probably have heard, except for a few pilots that we did on the analytical side, not the assay side, we really just chose one lot to begin with. So, and then there were questions too of like, we got the plant. Our colleagues at University of Mississippi helped us create an extract. But if we did either of those steps again, would we get something slightly different? And the answer is probably. And then how much would that affect the bioassay? So
Yeah.
that is a question of, we have a pretty well characterized library of constituents. Of course, we would all love to go deeper and know everything in there. But yeah, if we did any of this again, would we get the same? So that's why we're also trying to be very careful that we're assay focused and not, trying to walk away with a conclusion about milk thistle.
Yeah, thanks, Connie. I think there's a couple about oh, it keeps moving as I try to focus in on things like concentration determination and identification. I saw one that's too much data, which I agree is overwhelming at times.
But that's true
But
if you're
good to
trying to
have.
make a decision. That's the thing. We're trying to put these together and put these pieces together. But sometimes it can be hard or conflicting data.
Yeah. If you run a battery of 20 assays, what does it take to find a negative?
Yeah. I also appreciate human relevance is pretty big. I don't think that's specific for botanicals. It's a NAMS issue generally, but that doesn't mean we just ignore it, right? So that is something we're trying to, again, put these pieces together, think about ADME, think about concentrations, but what's actually getting to a human? And, is it always a healthy human that has the issues? Probably not.
Mm-hmm.
Lots
Right.
of
It seems like,
nuances.
yeah, seems like we've stabilized, but definitely human relevance, solubility, mixtures, chemical complexity, and standardization seem to be the big ones. And it seems like even some of the smaller font size are also tied into those bigger concerns. Thanks. All right, and then this one's more like a future directions for the BSC, so it's a ranking question. And so the request is that you rank the following areas in order of priority for follow-up and next steps. So one would be the highest priority. I'm going to guess, based on previous answers, ADME will be ranked fairly highly. Yeah.
Ooh, we might have a duel though.
I know, and lots of variability is really coming up in the ranking.
And one thing we talked about it in 2 of the presentations, but for neuro, for I think about 8 constituents for the MEAs, we compared individual constituents with the mixture in the same lab, same setup. And then we didn't spend a lot of time talking about today, but I think it's a really cool study we did with Axiom. We ran 80 of our constituents across the botanicals and compared that to the mixtures. So I think there's a lot of really cool things we've generated data on that we need to look at. But that's something we could potentially look at for other assays. For some that maybe aren't validated, sometimes it helps for that understanding, benchmarking single constituents with the mixture effects too, which might give us insight of, are these usable for botanicals as complex mixtures? The tricky thing is, as we talked about, it's not always one constituent driving it.
Yeah. All right, so I do feel validated that ADME ended up in #1, but it was quite a close race between that and the extracts versus isolated constituents. Seems to have stabilized, so I will move on to the next. Oh, we had one more. All right, I'll move on to the next question. So this is what's one key question or uncertainty that you would like answered before next year's meeting? No guarantees we will be able to answer it, but it would be helpful to know what you all view as a key priority for the next year or an uncertainty that you identified when listening to today's talks. And I think one thing with the ADME is because we largely have in silico information right now, it's for those individual constituents. And so comparing that back to the whole mixtures can be complicated, but maybe that's an area where incorporating individual constituent information would be helpful, like in the actual assays.
I think we can promise on the more dermal data as some of those are
Yeah.
getting going. So that's one I think we can at least say they're initiated by the time we meet next year.
Yeah. Identification of which assays are most valuable to screen.
Ooh, AI modeling. That's of course the buzzword, but if people have expertise with that, if we could throw a bunch of data and it could put things together, I mean, as a pilot, I think a lot of these would be, that could be cool. So if someone has
Yeah.
ideas on that.
And I think generally there's a lot of interest in like using like AI to integrate data from either different constituents and try to model like a mixture effect or from integrating the different toxicity assays themselves.
Yeah. Validate token incorporation into assessment framework.
Gut microbiota.
We have, I mean, in the tiniest bit, thought about that with like genotox in silico that you wouldn't necessarily throw just because it's not getting it absorbed. You still do have to worry about like gut tox. That's not really microbiota, but it's something of thinking in the gut. And then even if you have sugar molecules, the microbes there might be able to break off the sugar molecules. So you might have more active forms of the constituents. So maybe we could do more than that in the next year, but that is one thing that just came to mind when I saw that.
Yeah.
Ooh, nephrotoxicity. We wrote one paper, we got asked to write a paper that we had a really excellent postdoc at NIEHS led. And so we have a review paper on nephrotoxicity where we looked at assays and botanicals, but we haven't heard a ton of interest for that as a full group, but maybe now's the time.
Yeah.
Lots of having public, yes, that is one we really want to have so people can play these data, so data will be publicly available. Cool.
Well, gastrointestinal metabolism. In a
I
group.
definitely think some of these, like, yes, next year we will have. Some of these, it's like, have we considered them?
Yeah. So data integration, toxicity predictions, addressing human relevance.
I think that's really cool with the like what Catherine was starting to look at and some of the dose information.
Regulatory implementation.
We're not regulators, so that's up to y'all. But it is like, having these conversations with FDA, what's useful for them, what they want to see, and non-FDA, right? EFSA, Health Canada, others. So, we're trying to make the science available. That's why I threw my hands up at that. But, to be a little bit funny, but really we're trying to lay what's actually known about these. How can you use these tools? Do you need to make any changes? But then that piece, like we're happy, to speak about what we've learned with folks.
Yeah. We're gonna finish. All right, and so then this one is more just like general feedback about either the annual meeting, the content of the annual meeting, or the BSC in general. Thank you. I'm glad that the first comment was very positive. All right, for this one, I don't know if we all need to review the general feedback, but I'm going to leave the question open and just stop sharing my screen. So that way you all have an avenue to provide your feedback. And then if there's any additional discussion folks want to have, you can raise it in the Q&A or perhaps raise your hand.
Thanks, Julie. I think that gave us a really nice snapshot of, what we've heard today, what people would like to see in the future. I don't see any questions in the chat. I'm happy if anyone on the panel had any kind of cross-resonating thoughts or things they would really like to focus on or to see, let's say by this time next year. So maybe open up to the panel if anyone has any thoughts. I know it's lunchtime too. That's the tricky part. And late for some. Catherine might be doing her Guy Fawkes celebration already.
Oh, yeah. And hopefully Shane went to bed.
Yeah.
Yeah, not quite yet, but fireworks are starting to go off, so I will stay on mute.
Bill said, I would like to see studies include equivalent human doses to see relevance. Yeah, that's a really fair point. And when we started, we really did based on limit of solubility and then tried for as many as we could to have these long ranges. I think this is where others that know more about IVIG can say more, but maybe there's some where they're totally not getting in on the constituent level. We can at least say, throw those ones out. Would that change our call? Or is it something that, okay, we saw toxicity in the assay, but that's really not something we expect to see? So I think there's some things we can do with the data we have, but then in the future, yeah, is that something we do a little bit backwards, projecting on what's a human taking and then put that in there? Yes, Christine, come on. Go ahead, Christine.
That raises a thought in comparing our NAMs, our in vitro assays, with the human exposure situation, in that I bet most humans that are taking one or more botanicals are taking them on a more chronic basis, whereas what we're doing in vitro is an acute exposure situation.
Yeah.
So that adds another layer of complication to being able to correlate the assay dose levels with what humans will experience.
That's a really good point, Christine. I know, we mentioned very, very briefly for some of the cardiotox that some of the gentlemen there went up to more like 7 days. But is that even chronic, even though it's it's still in vitro, but some of these are very short exposures. So I think that's a good point. We're almost in this phase of can we do an acute screen, but that's not always going to capture some of the effects. Some of them you have to get to a certain level inside to see effects. So I think that's really worth raising as a limitation so far and something we can think about. Good question. Okay. Well, I know it's been a long day. We went through a whirlwind of data. I hope we can continue to share, exciting papers and things with you all. And as the data sets are made available, make sure people know and ideas, even when we make a data set public, there's still other analyses we can do with them. So we're going to do our best and within the groups decide how to package it. But I really encourage folks, if they have ideas for things we can do with the data we generated or things we could do moving forward, to let me and Julie know. I think with that, we can wrap up a little bit early. I really appreciate everyone's time. I appreciate our panel so much for walking through the different work. And I really just thank you all for your attention. And we will send the recording out in the following days. All right. Thanks, everybody. Take care.
Thanks, everyone.
Okay, thank you. Bye-bye.
Bye, everyone.
Bye-bye.